Hey everyone, welcome back. This is about the anaerobic respiration, which is also called as a fermentation. It is a Greek word which means that to boil. It is a Latin word, fermentation. Why does it is mean like fermentation as actually boiling? Here, Joseph Louis Gay-Lussac were the first one to identify the fermentation process. He was a French chemist discovered in the year 1815, demonstrated by the Johannes Wessel, which is also called as a Wilhelm Kolhen, in the year 1877, coined the term enzyme also. He was inspired by the Louis Pasteur, who first discovered the fermentation in the year 1857. Pasteur effect also we can call it. And here yeast contain the zymase enzyme, which is helping for the conversion of glucose to alcohol and also carbon dioxide. And here in this experiment we are taking the yeast as well as the a little amount of the sugar solution also, which is the 10 percentage, so that yeast is helping for the fermentation process. So when it is moving that, yeast ferment sugar to alcohol and carbon dioxide, and carbon dioxide gas collects, causing the liquid level in the upright tube to drop, so that carbon dioxide will be boiled, so that the drop will be happening. So when KOS solution is add a crystal of this, then which absorbs the carbon dioxide, making the liquid level to raise back up, confirming that carbon dioxide production is being there. This confirms that carbon dioxide is being released by the yeast during the fermentation process. So this is how the reaction starts. Glucose is converting into ethyl alcohol as well as the energy and to carbon dioxide. So when we see the equation alcoholic fermentation, it is being taken by the Saccharomyces cerevisiae. Incomplete oxidation is achieved under the anaerobic conditions. Pyruvate is converted to acetaldehyde in the presence of pyruvate decarboxylase. Thiamine pyrophosphate Mg2 plus been added, and then acetaldehyde converted to ethanol by releasing two molecules of ATP energy, and by the enzyme alcohol dehydrogenase by taking up the NADH2, which has been produced in the glycolysis process to become the NAD plus. Even lactic acid fermentation, if you see, bacteria produce lactic acid, and even in animal cells during the exercises, incomplete oxygen will be present, so that pyruvate can convert it into lactic acid also. In both lactic acid and alcohol form, not much energy is released, less than 7% of energy being released, and yeast poison themselves to death after reaching to the 13% of the alcohol. Remaining other enzymes will continue the process of fermentation. Thank you for watching.